gas came from its mobile phase that is used in the gas chromatography. So in the gas chromatography, mobile phase is the gas, that is the different carrier gases are used as a mobile phase, that are hydrogen, helium, nitrogen, etc. So gas chromatography, it is a partition chromatography in which we can separate out different compounds according to their boiling points. Mainly the volatile gases that can be separated by using the gas chromatography. So we will see it in detail. So firstly, here is the definition of chromatography. So chromatography is defined as the physical method of separation in which the mixture of analytes is separated using two phases. One of them is the stationary phase and another one is the mobile phase, which percolates through the stationary phase. The separation occurs because of difference in the affinity between analytes and stationary phase. And here is the principle of gas chromatography. So as we know that the gas chromatography, it is a separation technique. So this is the principle. Gas chromatography is based on the principle of partition of an volatile compound in two phases, a liquid phase covering the adsorbent surface and a gaseous phase of the eluting gas. So here are the two phases. Stationary phase is the liquid and the gaseous phase is the gas. And with the fixed set of parameters, we can do analysis like the length and the diameter of the column, temperature of the system will be maintained, nature and the flow rate of the eluting gas is important. So this is the principle of the gas chromatography. Now we will see the gas chromatography block diagram. So first part of that is the gas cylinder. As we know that the mobile phase or the carrier gas is the gaseous phase. So we want the gases like hydrogen, helium, nitrogen. So first one is the important part, that is the carrier gas or the gas cylinder. After that, from this regulators, this gas will enter to the injection port, and in the injection port we can inject the sample via the micro syringe, and the capacity of the micro syringe is the microliters. From this syringe we can inject the liquid sample into the system. So when we inject this liquid into the injection port, it will get converted into the gaseous form due to the column is kept inside the oven and the temperature of the oven will be higher. So this liquid sample, when enters into this column, it will get converted into the vapors and it moves through this circular column or the spiral column. The length of the column is very large. It varies from 2 meters to the 50 meters and the diameter should be in the centimeters. This column is made up of the material which withstands for the higher temperature, like the thermosteel, then the Teflon and the glass materials. The material of the column that will be inert, it cannot react with our sample material. So the sample will move through this spiral column. It then lastly enters to the detector. And in the gas chromatography, according to the boiling point, compound gets separated. So firstly, the low boiling point containing compound will come to the detector, and then the higher boiling point compound will come to the detector. So according to that, the detention time of the sample will be different. So low boiling point compound comes firstly to the detector, so its retention time will be shorter, and when the higher boiling point containing compounds come to the detector, it requires larger time, that is the retention time is more. And here the various detectors can be used like the ECD, FID, TCD or the photoionization detector. So in the gas chromatography, 4 kinds of the detectors will be used, and when our sample reaches to the detector, there it will get measured, and after that the signal will be measured or the recorded on the recorder. Whole instrument, that gas chromatography, is computer controlled. So this instrument is known as a chromatograph, and the spectrum which is observed on the recorder, that will be called as a chromatogram. So both terms are different. Instrument is known as, or the whole assembly is known as a chromatograph, and the spectra which is observed on the instrument, it is called as a chromatogram. So according to the boiling point, the compounds will get separated and we will get the peaks on the instrument. So when we see the data in instrument, we will get the idea about which compound is present and the peak area, peak height, then the percentage area, number of peaks observed. That data will be observed on the software. So this is the block diagram of gas chromatography. Again, here I have written how the gas chromatography works. As a sample is injected into the injection port, sample will get vaporized. It is forced into the column by the carrier gas, that is our mobile gas, which is usually used helium. In some instruments, hydrogen will be used. Because of its explosiveness, it is rarely used. Then components of the sample mixture interact with the stationary phase so that substance take different amount of time to elute from this column. The separated compounds pass through the detector. We will get the electronic signals collected over the time, that is the detention time, and that will be sent to the gas chromatography software, that is GC software, and the chromatogram is generated on the recorder. So this is all about the block diagram of a gas chromatography. Now we will summarize here the components of the gas chromatograph. As we have seen in the instrument, first part is the gas cylinder, that is the gas supply is important because it acts as a mobile phase. So here nitrogen, helium, that will be used as a carrier gas or the mobile phase. Sometimes hydrogen will be used, but due to its explosiveness, the helium and the nitrogens are much preferred. Then the sample injector. Here the sample injectors are the micro syringe or the rubber septum. And then the column in the gas chromatography, capillary column and the packed column, these columns are used. So here the gas columns, which are the packed columns. So these are tubing packed with small uniform size inert support coated with thin film of the non-volatile liquid. So inside the column there is a non-volatile liquid that will be inert. It will not get easily vaporized at the temperature of the oven. So this kind of the non-volatile liquid that will be used in the columns. And last part is the detectors. So here in the gas chromatography, 4 detectors are mainly used, and each detector can be used for the specific compound analysis. So first one is a thermal conductivity detector. The second one is a flame ionization detector. Third one is a electron capture detector. And the last one is a photoionization detector. So these 4 detectors are used in the gas chromatography, and each detector is useful for the different kind of compound analysis. So by using gas chromatography, we can do qualitative as well as quantitative analysis. So in the qualitative analysis, we can find out the purity of the organic compounds. Is there any contaminant present or not, that can be revealed by using the additional peaks which are observed in the chromatogram. Then we can find out the areas under the peaks, and that will show us the contamination is present or not. Then this technique is also useful for evaluating the effectiveness of purification procedures. Then from this chromatogram we can find out the retention times, and retention times gives us idea about the identification of the components which are present in given mixture. Then the gas chromatography provides an excellent means of confirming the presence or absence of a suspected compound in a mixture. So by using gas chromatography, we can do the qualitative analysis. Now we will see the quantitative analysis. So in the quantitative analysis, we can measure the peaks with an accuracy, and this data gives us the reliable results. Now here are some advantages of the gas chromatography. So gas chromatography requires only very small samples with little preparations, because the sample which are injected into the machine that are in the microliters. Then gas chromatography separates the complex mixtures into components. Then results that can be rapidly obtained. The time required for the analysis is in between 1 to 100 minutes. Then the gas chromatography instrument gives the precise results, and only instrument with the sensitivity to detect the volatile organic mixtures of low concentration. So this is an use of the gas chromatography, that we can separate out the volatile components or the low boiling point containing compounds with low concentrations on the GC. And this equipment is not very complex, but the oven is sophisticated. So this is all about the gas chromatography. Gas chromatography is the technique in which we can separate out the compounds according to their boiling points. The gas chromatography technique is applicable only for the volatile gases or the volatile components. So thank you.